Hi, everyone. Welcome back to day 6 tutorial. We've now talked about the theory and application of dynamic causal modeling. In the fourth part of our teaching for today, we're going to explore dynamic causal modeling for fMRI data more deeply. So we're going to first start with an extension of DCM for fMRI to connectomics. This will be presented by Peter Bedford, who's a research analyst in my team. And then we're going to go through some kind of practical simulations, some hands-on learning by Povilas Karvelis, a postdoctoral fellow in my group. So I'll hand it over to Peter. Take it away.
Thanks very much. So there have been many variations on DCM, dynamic causal modeling, since it was first introduced in 2003. Today we're going to focus on one variant called Regression Dynamic Causal Modeling, or Rdcm, introduced in 2017, which we happen to use here in Professor Diaconescu's lab. So if you were in the last lecture, you might remember DCM considers changes in regional brain activity to be influenced by exterior disturbances, which we label and by the activities, which we label X, of the other regions in the brain. On this diagram, I've omitted the bilinear interactions. You can see the state equation here, no bilinear term, because these won't be included in our DCM. So these dynamics are encapsulated by this state equation shown in the green circle. And DCM posits that these network dynamics can be inferred from bold signal data. So here's that state equation again. Notice that to find the parameters A and C, we essentially need to solve a system of differential equations. This is computationally rigorous even for linear DCM, especially in cases where more regions are needed and where bold time series are longer. For this reason, as you've seen, there's a lot going into the choice of regions and final network architecture, from developing an informative experiment to defining a well-informed hypothesis to Bayesian model selection. So while DCM works under the correct conditions, it would be nice if there was a way to avoid these constraints, certainly for situations in which no well-informed hypothesis can be made. Rdcm addresses this limitation of DCM. While Rdcm and DCM provide models of comparable quality, Rdcm is much more computationally efficient. With the same computational burden, Rdcm is capable of modeling networks with over 100 different brain regions. And the main import of this computational efficiency is the opportunity to take a more exploratory approach to analysis and interpretation of effective connectivity, since a whole brain picture can now be available. So how is this improvement possible? Roughly speaking, Rdcm is a modification of DCM by opportune use of approximations and choice of coordinates. The transformation from DCM to Rdcm takes 4 steps. First, we transform the bold time series, the frequency domain, along with the state equation we already saw. Second, we linearize the hemodynamic model. Third, assume partial independence of the connectivity parameters, or in other words, use a linear DCM. And lastly, we implement a gamma prior for noise precision. Let's take a minute to look at these steps in slightly more detail. So the first step is to transform the state equation into the frequency domain. Notice that the system described in the final line is still linear with respect to the model parameters. If you're not familiar, this middle line is, the last two lines are a property of the Fourier transform, which basically states that the derivative is a linear multiple of the function itself. The second step concerns the observation model, the link between the hidden neuronal dynamics to the bold signals. This model involves nonlinear differential equations, which describe hemodynamics, and a nonlinear static bold equation. To make translation to the frequency domain more straightforward, we'll simply assume a fixed hemodynamic response function and convolve it with the bold series. This is equivalent to multiplying the functions in the frequency domain. These are continuous representations, continuous functions, but our data will inevitably be discrete. So we need to account for that, and we need to account for the noise which affect the MRI data. So we discretize the frequency and time into points indexed by the vector m, you can see here, and introduce the difference operator of the discrete Fourier transform by making the linear approximation to the exponential function. So now we need to account for measurement noise. And like in classical DCM, we assume white noise with distinct variance for each region. And the dependence of the noise vector on the endogenous connectivity parameters, or the A matrix, is actually going to make derivation of the likelihood function difficult. So to get around this, we need to assume that the noise for a given region will not depend on parameters describing connections between other regions only. So that's where we're assuming partial dependence, as well as the disregarding bilinear terms in the state equation. So this is equivalent to a mean field approximation, and this makes it so the posterior can be factorized along sets of connections. And this is exactly what accounts for the increased computational efficiency. So with all these changes, we have a new state equation for Rdcm. Notice that it's now a regression problem rather than a differential equation. In effect, we've reformulated a linear DCM in the time domain as a general linear model, or GLM, in the frequency domain. Last step would be to turn this GLM into a generative model, and for that, we'd need to specify priors for the parameters and hyperparameters. Again, following classical DCM, we keep zero mean Gaussian shrinkage priors for the connectivity parameters, but we need to depart from DCM by choosing a gamma function as the prior on the noise precision. And again, that's just going to simplify the derivation of the posterior. From there, the parameters and hyperparameters are derived through the variational Bayes technique, which searches for parameter and hyperparameter values, which lead to a maximum negative free energy value for the system. So in principle, very similar to classical DCM. Even though networks of more than 10,000 connections are possible, it's rarely the case that all of the connections are really important. And it's often the case that some pairs of regions don't influence each other at all. So to reflect this fact, we have two methods of implementing sparsity in the network. The first method, which I will name the structural prior method, involves using information about anatomical connections to construct the prior for the connectivity matrix. In other words, we fix the prior for a certain connection to 0 if we have structural MRI evidence that the two regions are not anatomically connected. The second method, which I'll nickname the sparsity optimization method, involves using another slightly altered version of Rdcm, which includes extra hyperparameters, which represent the probability that a given connection is actually present. So in this case, the Rdcm inversion procedure finds these probabilities through the variational Bayes technique, just as it finds the other hyperparameters. Then we would prune the connections which have a low probability that they are truly present. So in Professor Diaconescu's lab, we're using Rdcm to investigate differences in whole brain affective connectivity between LSD and placebo conditions during resting sleep. As some of you might know, LSD is now being studied as potential treatment for major depression disorder. We connectomics researchers wonder whether the efficiency of efficacy of LSD as an antidepressant treatment could be predicted by looking at patients' brain connectivity. One route to answering this question would involve first looking at whether the different subjective effects of LSD can be predicted from brain connectivity. But before that, we ought to see whether we can correctly classify LSD or placebo using brain connectivity as classification features. We have attempted to answer this preliminary question by analyzing whole brain bull data from a resting state fMRI experiment in which patients were measured twice, one time after taking LSD and the other time after taking placebo. As I describe our analysis pipeline, you might notice that it resembles generative embedding if you happen to be familiar with that method. So the imaging data provides the input from which we compute whole brain effective connectivity using Rdcm, and we're trying both sparsity implementation methods. Then the region-to-region connection strengths will act as features on which we'll train machine learning classification models, which classify the connectivity datasets by condition as LSD or placebo. Then using cross-validation, we can gauge the accuracy of these two classification models and compare their performance. So in this experiment, the subjects were not asked to do anything specific while they were measured. Because the data was taken during a resting state, we do not consider any experimental inputs. In this case, the only parameters of interest are the endogenous connectivity parameters, or the A matrix. So here we have the top 500 connections by mean effective connection strength, estimated through the structural prior method for LSD on the left, placebo in the middle, and the difference between the two on the right. These plots show connection strengths averaged across directions. So in other words, the connection shown between region A and region B is the mean of the connection directed from A to B and the connection directed from B to A. So it's also worth noting you can't actually see any self-connections from the connectograms. There's no connect line from one region to itself. The strength of the connection is indicated by the line thickness and opacity, while the sign of the connection is indicated by the color. We can see that effective connectivity generally increases under LSD, since they're among the top 500 are mostly red colored connections in the third figure. But there are some exceptions, most prominently bilateral connections in occipital areas. And these results are echoed by the sparsity optimization method. As can be seen from a paired t-test comparison, many of the most different connections are indeed between visual areas. But there are some important connections that don't involve visual areas, like the self-connection for the anterior cingulate gyrus and the connections to subcortical regions. So those ones would be highlighted in a gray box. The stars are labeling self-connections, which happen to be the only negative connections. And these ones have the opposite directionality on this plot from the positive connections in terms of their relative magnitude. So similar results can be seen from self-connections. So on this plot, there are only self-connections, which roughly represent the activity decay rate for a single region. So if a region happens to have high in magnitude self-connection, but the connection is negative, that means that whenever the region itself increases in activity, it will itself start to decay faster. And if it has a low self-connection strength, then its decay rate will be slower. So we found that the classifiers trained on actually functional connectivity and these two versions of effective connectivity yield comparable performance, which was pretty high at around 90 to 94% balanced accuracy. Neymar's test indicates that there was not a significant difference between the performances of the three methods. So even though using effective connectivity didn't improve classification performance over the more traditional functional connectivity, the fact that it was comparably effective supports the validity of the approach. Okay, so this concludes my portion of the lecture. Thanks to everybody who supported the research, and thanks to everybody for listening.
Okay, thank you very much, Peter. There are no questions yet in the ask question, nor in the chat. Yeah, we can see if some questions come up. I have maybe a couple of just conceptual questions. So in the context of so one of the advantages of using regression DCM is that it allows us to perform exploratory analyses. From your perspective, sort of what is the benefit of using regression DCM, which assesses effective connectivity, compared to other functional connectivity measures such as ICA or seat-based connectivity? What does regression DCM and effective connectivity add to our understanding, for example, of LSD effects on connectomics that is lacking in functional connectivity approaches?
Well, the main difference between effective connectivity and functional connectivity is the lack of directionality in functional connectivity. So in effective connectivity, you've got this parameter, this connection strength, which gives you a sense of how much one region influences another. And for functional connectivity, there's no difference in the amount that one region A affects region B as region B affects region A. So with effective connectivity, you can have an imbalance you can, in fact, sort of see a kind of hierarchy of brain regions represented, a direction in terms of a chain of influence, and not only that, functional connectivity considers that regions do not have an influence on their own activity, or rather, their influence on themselves is statistically, 100%, basically. Whereas effective connectivity actually leaves room for a little bit of difference between the regions and how much they affect their own activity. And we saw that with the self connections. So there may be some regions that are more inhibitive of themselves in one condition and less inhibitive of themselves in another condition. And this might add a little bit of insight, especially in this case where the anterior cingulate was one of the most highly different self-connections between the two regions. So considering that the difference of the conditions was one was on LSD and one wasn't, that might be tied to kind of the difference in self-concept or error, I guess, integration, error handling, which has been, kind of reported as a symptom of LSD. So that's going to be part of our, forthcoming research is breaking down what do these differences in connectivity actually mean in terms of differences in symptomatology, or in the case of LSD, it would be more like subjective experience.
Okay, great. There's another question from John, and I just invited him to come up and ask it himself. But I can read it out loud in the meantime. How do the Rdcm effective connectivity changes compare to the equivalent comparisons in functional connectivity in these analyses? Are there overall topographic patterns that are highly similar?
Yes. So
hi, John.
the similarity.
Hey, thanks for the invite. I have another question as well, seeing as you invited me up, so I'll jump in on that one after.
Okay.
See if I had to go this one.
Okay, so to address your question, we don't have right now a formal understanding of how different they are between using functional connectivity and effective connectivity. But if I was to show you a connectogram plot for functional connectivity, just like the ones that I showed for effective connectivity, the magnitudes of the connections, so not their color, right? There wouldn't be a color difference in functional connectivity, but their magnitudes do follow a visually similar pattern. There's the prominence of that those bilateral connections between the occipital regions, and the same difference between the two conditions. So generally, an increase in functional connectivity under LSD, but specifically a decrease for those visual regions. So we actually, part of our scope, I think, is to maybe compare these two methods, functional connectivity and effective connectivity, a little bit more formally, to compare, how do the rankings of these magnitudes of connections stack up to one another. But right now, we just have kind of the visual result.
Yeah, great. I mean, that was what I was wondering, just phenomenologically, like the fact that you see similar patterns, that's very reassuring, because it would kind of be a bit of a concern if you, given kind of how much the results should be kind of driven by the data, right? And you're quite close, kind of in the linear domain, so like you're not that far away from what the actual correlations are. So think like the interesting question is not so much a question I asked, which is like how similar are they, or are they similar, but more like in what ways do they differ, right? Which I think is what you were suggesting you're going to be taking a closer look at. So that's great. I had another question. I mean, feel free to just kind of pass on this one, but I was curious that because you were going through the derivation a bit, which was great, and you kind of in the Fourier domain for a while, right? Kind of move into there, and then other things get simplified. So what I didn't catch from there was, are you kind of broadband frequency? Like, what is the frequency range, right? Because normally, like you would, well, often when you go into the frequency domain for functional connectivity, you do frequency band binnings, right? And say, okay, this is my kind of 10 frequency bands or 5. Or you just filter with one specific frequency band. You know, a lot of functional connectivity fMRI modeling with kind of Kuramoto oscillators. They have a pretty narrow band, but they kind of filter the fMRI signal, and you get kind of quite a strong oscillation when you do that. So are you, how does that relate? Like, are you kind of is this explaining the entire power spectrum? And then like when you coming back to the first question is like, if you're going to do comparisons with the functional connectivity, then the natural thing to do, I think, in addition to looking at the Pearson correlations, would be to look at different frequency bands, because I think this model formulation involves kind of broad frequency band complex covariance terms.
Yeah, I don't know. I'm not exactly sure. If I can address that. I mean, I assume what you're saying is what is the range of frequencies, because I heard the word binning, but I think the frequency spectrum is discretized according to sort of matching obviously the time number of time points, but also use the repetition time in the transformation as well. So beyond that, I'm not exactly sure. Can you still see my screen?
You've disappeared now for me, actually.
So
no worries.
Okay, thanks a lot. So I think we can move on to our tutorial that will actually allow us to simulate some bold signal data. So I'm going to bring Povilas to the forefront. Thank you so much, Peter. And Povilas, would you like to share your screen?
Absolutely. Thanks for bringing me up. Let's see. All right. There we go. So can you hear me okay and see everything fine?
Yes.
All right, good. So I'll start with the kind of quickly, with a quick overview of
maybe
what
you
we're
can
gonna
just
do.
hide this little window that appears. Yeah, perfect.
Yeah, there we go. Yeah, so I'll first start with a little overview of what we're gonna do, and after that we'll jump into the code and go through everything step by step. So the goal of this tutorial is to introduce you to some kind of basics of dynamic causal modeling for fMRI data. Today you've heard just now about more advanced methods, and earlier you heard a lot about DCM in a lot more detail. So this will be perhaps a little bit easier as a kind of an introductory exercise. In this tutorial, we will consider this experimental paradigm, again, something you've heard about earlier today, where the subject is inferring on the intentions of others. So in the sequences of trials, a subject receives advice, and then that advice can either be correct or incorrect, or either helpful or unhelpful, and therefore the subject keeps track of how helpful the advice is and forms some kind of expectations about the advisor's fidelity, and that's what we'll be interested in, or I guess that's going to be an important part in our modeling. Specifically, the advice prediction error. Here we have epsilon2 in this model, and the model itself, we're not going to go into too much detail. The previous tutorial covered this hierarchical Gaussian filter in a lot more detail. Here we will take this output and use it for DCM simulations. So that's kind of the context, just to, make it, I guess, give it some kind of color and context for the coming simulations. So the prediction error that we're interested in the previous research has been associated with certain regions in the brain. So here's a screenshot from a paper that Andrew published a few years ago, looking at the regions that encode this prediction error. And among many regions, we have dorsal medial prefrontal cortex as involved in it. Also temporal parietal junction. Here we probably can't see it that well. And these two regions, these two regions are the ones that we will be focusing on. So this example is a bit of a toy example, I suppose. The actual findings and the actual story about this, how the brain encodes these prediction errors and which regions are involved is a bit more complicated, but for the purpose of this tutorial, we're going to consider how the temporal parietal junction and dorsal medial prefrontal cortex are involved in the context of updating your beliefs or some kind of surprise when advice is either correct or incorrect. So in this simple schematic, we have basically the model that we propose. So in this tutorial, instead of using real data, we will be simulating data. And the model that we have here is as follows. So we have two nodes. X1 corresponds to temporal parietal junction, and X2 corresponds to dorsal medial prefrontal cortex. And then we have a stimuli that is our input. And the stimuli here would be simply the instance of, either correct or incorrect advice. And then the context, U2, which modulates the forward connection from X1 to X2, would be the prediction error that we get from this hierarchical Gaussian filter. And in matrix notation, this is how it looks like. Again, that's the equation you saw earlier for describing the dynamics of, or the neural activity of these different nodes. And here we specify different parameters that we're interested in. Particularly as it relates to the prediction error, we have this parameter here that encodes the forward connection from x1 to x2. For the driving input, we have C parameter here that only takes into account the U1 part of the input, so the device itself. So that's kind of our equation for this model. What we're going to do next, so first we're going to simulate some data with this model, and then we will use that data as if it was actual data that we collected, and we did not know which model or what is the exact mechanism by which the data was produced. So we will fit different models to test these hypotheses. The first model is going to assume no modulation, where we only have the main driving input and no modulation. A second model will assume that the prediction error modulates the self-inhibition of dorsal medial prefrontal cortex. And then the final model is going to be the model with which we produced the data. And so by fitting these, we would expect, if everything goes well, that we would find that this model is the most likely model that produced the data, because that's the model with which we simulated the data. So this exercise can help us to kind of establish some kind of face validity of the models. It allows us to test certain assumptions or certain constraints that we need for our experiment to be able to find the model that actually produced the data. But of course, especially in the example that we're using, it's a kind of a co-example, and therefore it doesn't really tell us how, let's say, valid it is as a construct in terms of real life situations. So it might tell us that in principle, given the constraints or given the assumptions that we put, we would be able to recover this kind of model. But I guess in this line of research, there would be other steps for further establishing the validity of the model. So the second step would be construct validity, where you actually use the collected data and that way try to validate your model. And then finally, you have predictive validity, where you would predict additional features of data based on what your model suggests, and then test whether those predictions are true by conducting experiments. So this is kind of the context. We're kind of mostly focusing on some basics of DCM, but there are also these more kind of general aspects of data analysis when it comes to simulations and how to think of the value of this analysis. So let me now jump into the code. There we go. So for hopefully most of you have already been in previous tutorials and know how this works. But just in case, the first thing to do is to make sure that your directory is in day 6, and you might want to run KCNI set up paths to make sure that you have the right paths set up for this tutorial. Sometimes takes a while for the paths to be set. There we go. And once that is done, move to DCM for fMRI tutorial directory. And the main script that we will be working with is called DCM compare demo. So that's our master. This is our master script. There's a brief kind of summary similar to what I just presented of what we're going to do in this tutorial. And so the first step for this analysis is to so let me just first all the previous variables in the workspace. So the first thing is to run. We want to generate some prediction errors, and that we can do by running this section here. So you can that's the wrong shortcut. You can run this section by section, and in this part, this is basically shows what we generated with this part of the script. And as I mentioned earlier, this is not too essential for our purposes. Basically, what we have here is the we decided to generate advice that it starts out as correct most of the time, so it has high probability of being correct. And then after 100 trials, this changes and it's mostly incorrect. And then by using this as an input into our hierarchical Gaussian filter, we can produce, we can estimate the prediction errors that the subject would experience by receiving or by seeing this sequence of advice. As you can see, there's a very sharp, very large prediction error here, mostly because it comes after a long sequence of correct advice. The subject gets incorrect advice, and therefore that's very, very unexpected. If you wanted, you can go inside the script and look into more detail.
Povilas?
How this, yeah.
Just sorry to interrupt. Maybe you can increase the font size a little bit by going to Home and going to Preferences. And then there's fonts, yeah, and 12 maybe is good. Just
there we go.
Yeah, that helps a little bit with readability.
All right, yeah, thanks for that. So yeah, where were we? We're in this script where we generate advice and fit the model. So perhaps one thing to mention here is that the way we generate prediction errors is based on running the model with parameters that are informed by what has been found empirically in this kind of context. So we get something quite realistic here in terms of the prediction errors. And then the next step in our analysis is to use this output, which contains the prediction errors in the model that we want to generate. So dynamic causal modeling. And what we do here is first we set that there's two regions that we're planning to simulate. We set the names of the regions, and perhaps the most important part here is we decide on the connectivity matrices and what kind of connectivity we want to have when it comes to the endogenous or intrinsic connectivity. So here we assume backward and forward connectivity from X1 and X2. Then we assume that the prediction error is going to be modulating the forward connection between X1 and X2, and that the driving input, or U1, is going to enter at X1 location. So that's, we have two nodes, and therefore our specification of the model is pretty straightforward. And once we do that, the model that we specified and this output from that contains prediction errors, we feed this into this function, and we can run this segment and let's see. I think I did it twice, so there's going to be a brief lag. All right, so my plan was, while it's running, to go inside this script and have a quick look at what it does inside. There we go. Right. So the first thing to do for trying to model fMRI data is to space out the trial. So in the previous part so let's minimize this. In the previous part where we generated the prediction errors, we just had 200 trials, and now when we want to simulate fMRI data, we need to decide how we're going to space them out. And for fMRI, due to its time resolution and the hemodynamic response and delays, there's an optimal way to do it. So here we're basically spacing it out quite a lot. For this toy example, we assume that each stimulus is each trial is going to last for 10 seconds, and there's going to be 100 seconds spacing between the trials. So that's what we do here. Then we specify all kinds of priors for the DCM for each of the matrices. We initialize some values, and when it comes to the simulations themselves, we run two loops. So the first loop is for kind of with finer steps, where we want to simulate neuronal data. So we want to sample it more frequently. And the second loop focuses on kind of the bold response, and therefore we sample less frequently, something that will be more realistic in real life. So each scan here happens at every 2 seconds. What else is there to say? So then the simulations themselves happen within the script, which is written to generate synthetic data based on the DCM specification. Takes as the input the model and also the signal to noise ratio. So here, as you can see, we have 1 for the signal to noise ratio, and that's kind of the default value. If we go further, you can, just like a rabbit hole, you can go endlessly into scripts. Oh, I think I missed it. Yeah, there we go. So somewhere in there, what we have is the state equation for the dynamic model responses, and here we have the equation that Andrea presented today that considers all these different variables when it comes to the hemodynamic response and how the neural firing relates to the signal that we can measure. So we have the excitatory neural activity, vascular signal, cerebral blood flow, venous volume, and so on. Right, so that's something that you can, as I mentioned, explore endlessly. A lot of these things we don't manipulate. So there was a question about, how these hemodynamic or forward model parameters are estimated or focused on. And as Andrea replied, most of the time they are not of interest and are not carefully examined unless people specifically focus on understanding those, I guess, parameters or that model in more detail. So this is our, that's the part where the neuronal and bold data are generated. So neuronal data is then, convolved or with the hemodynamic response or with this forward model and produces bold data. And yeah, the rest of the script is basically plotting these results. So now we can go back and take a look at what we produced. So I'll just close two of these figures because I produced them twice. Okay, so this is our first plot of the neuronal activity. And what we have here, so the plot up here is our stimulus or our inputs. And our first input is U1, and that's the red dotted line here. And this is basically comes from the fact that we had 10 seconds for stimulus presentation and then 100 seconds spacing in between. So that's our kind of inputs throughout the whole, I guess, scanning session. And as you can see here, even though we had correct or incorrect input or advice, here we model it as just as an input without distinguishing between correct or incorrect. And another of the inputs is the prediction error. And here, as you can see, it changes from trial to trial. And if I go back to the previous figure where we had this prediction error here, it's basically replotted just instead of a line, it's a scatter plot. It's replotted here with this, largest prediction error around this time point. And then based on this input, we have the neuronal activity plot. And in green, we have the temporal parietal junction. And in blue, we have the dorsal medial prefrontal cortex. And as you can see, the activity kind of mirrors this, at least in the dorsal medial prefrontal cortex, that activity mirrors the prediction error because that's what modulates the strength of activation of that region. So it's quite intuitive to see what we're seeing here. Now, the next plot shows us the bold signal. So this is basically the neuronal activity that we just saw converted into bold signal through the forward model. And as you can see, it looks quite noisy. And with looking at it with the naked eye, you can really tell what's going on that well. We can kind of see this, signal in the noise for the largest prediction error. It's quite apparent, but for the rest, it looks just noisy. So let me minimize this. let me go back to the main script. So, that's the data that you saw. And now what we're going to do, we're going to pretend that this bold data, this one right here, we're going to pretend that this is what we collected from some subject in an experiment. We want to try and find out, what is the best model to explain, the underlying mechanism that generated the data. And so here we are going to specify 3 models, as I described earlier. The first model is our null model that assumes no modulation. So here, the way we specify it is basically by switching on or off certain parameters or allowing them to be, estimated. So for the A matrix or the intrinsic connectivity, we assume that the forward and backward connections should, have some kind of non-zero values. For the modulatory matrix, we assume 0, so there's no modulation. And then for the C parameter, we assume that it's going to enter at, x1 or temporal parietal junction. So this is, our specification for the model, and then we feed this into a script for estimating the model, and then we save the output for later model comparison. And then we kind of repeat the same process for model 2. So here we have prediction error that modulates the self-inhibition of dorsal-medial prefrontal cortex. So the A matrix is the same. The B matrix is now different because for the diagonal, the last variable or the last element in the diagonal, we set this to 1, so we allow this to be estimated in the model. And then the C is the same. And then finally for model 3, which is our actual model, we set the modulation of the forward connection as the one that we kind of expect to take a non-zero value. So if we run this whole section, so here you can run it. Let's hide this one. You can run it by clicking run section, or there's a shortcut. I use Control-Enter, at least on PC. I think it might be different on other devices. So, right. So as it is running, as you can see, there's a recursive way to fit the models where we. Okay, so this is stuck for some reason. Perhaps it's gonna get unstuck, where the parameters are fit recursively, and the time series that we have here, it's kind of hard to see, but keeps improving the fit with each iteration. So that's the feedback that we have from this plot, and at the bottom we have some kind of information about the parameter estimate. So this is basically parameters starting with our intrinsic connectivity matrices to modulatory matrix, and then the C, and also the hemodynamic response parameters, some of which we, as Andrea mentioned, we kind of estimate as well at the same time as we estimate everything else. And the bars are basically showing the difference between the priors that we set, or the mean of the priors, and the current estimate or the posterior estimate of these parameters. So by looking at this, we can see how far off we are from, or how far our priors are from the actual estimates that we find. Right. So normally this is 10 times faster, but now it's going really slowly, probably because I'm sharing the screen. Hopefully it's not going to take too long. Maybe I'm just going to kill this right now, just because I've already run this part of the script earlier, and I have these, each of the model fits saved. So if you're doing this in parallel with me, probably you already have this part completed. And so now, once we have all of these model fits, we run model comparison settings. So this basically loads these three models or model fits that we have, and then we feed it into this interactive tool that will allow us to compare the goodness of the models. Seems like it's also going to take extra long for this one. Right, so because we kind of preloaded all of the settings, all we really need to do once this opens is just to click Run, and this will perform a Bayesian model comparison. So click Continue. Right, so as you can see, we get the results. So the order of the models here is the same as we specify them. So the first model was the null model. Second one was where we had self-inhibitory modulation of dorsal medial prefrontal cortex by prediction errors. And the third model was the actual model that produced the data. And as you can see, this seems to have the best log evidence, and that translates into very strong probability for this model producing the data. So that's normally what we want to see in this kind of analysis, that we are able to recover the model that produced the data. And another thing that we could do, and now I'm wondering if this is going to work given the slowdown of my MATLAB. So here I prepared another model, and you can play around with this and set all kinds of values that you want here for generating the data. But here we could consider one of the models that we used for fitting the data, which was modulating the self-inhibition of dorsal medial prefrontal cortex. So this is basically, everything is the same as before, only that instead of the forward connection modulation, we're modulating the self-inhibitory connection. And if we, let's say, run, okay, so I don't need to highlight. So if I just run this section now again with this different model, we will be able to see, there we go. We're able to see that we get very different neuronal dynamics. I guess the clearest effect is seen in here again. So with the with the largest prediction error, At this point, we see the largest decrease in the neuronal activity in dorsal medial prefrontal cortex, and that's because the self-inhibitory connection is inhibitory, and therefore prediction error reduces the activity. We can, increase this, for example, the strength of the self-inhibition, just to see the effect a bit more clearly. So yeah, it's a bit stronger. And again, this is our bold response related to that neuronal activity. And what we could do now, again, now that we simulated data with a different model, is to rerun this part of the script where we, so this, if I just rerun this, hopefully it's going to get fast enough. If we rerun this, it's going to override the previously saved models. And normally when this runs quickly, it's not that much of a problem. Now that it runs slowly, you probably would want to have save it under different names. Okay, so it's much faster now. It should work.
Povilas, I was sorry to interrupt. I was wondering whether you also included the backward connection in A, basically going from the DMPFC to the TPJ.
Yes, there's a backward connection and a forward connection.
Okay.
Yeah,
they're both included in there. Okay.
Yeah. Yeah, so this is, so here we are changing the the model that generates data. And again, there's all kinds of, settings or parameters or assumptions that we have built in. So even things like the length, or the number of trials, or the strength of, let's say, the prediction errors could also affect our ability to recover the actual model that generated the data. So in general, when it comes to this kind of analysis, which I guess in many ways is more general than just DCM, any kind of simulation analysis of a model where you then try to recover it, that way you can test all kinds of constraints under which you would still be able to recover the model that generated the data, and that can inform your experimental design and can reveal sometimes that you're actually not able to determine which model generated the data if you don't include specific constraints. So in many ways, this is a very, I guess, time efficient and resource efficient way of approaching all kinds of questions or hypotheses that you have. And again, this relates back to this kind of phase validity and different types of validity. So by running these kind of simulations, you can at least determine whether in principle you will be able to answer your question with the specific constraints that you assume for your experiment. Okay, let's see. So that's the old plot. I think that's the current one that is still running.
And maybe just to clarify to the participants, this SPM_FX, that FX fMRI that specifies the neural state equations which we've looked at in the theoretical part of the lecture, and the SPM_GX specifies the forward model, the observational model, or the hemodynamic response model that is used for fMRI.
Oh yeah, that's right. I think I might have slightly confused people earlier.
No, I think you mentioned it correctly. I just want to reiterate that the scripts themselves might be very daunting to read through, but essentially the aim is to implement the neural state and hemodynamic response equations, respectively, in these different types of models. So I was wondering, is there any point basically where this model comparison or model distinguishability actually breaks? Povilas, did you find it any point where you cannot actually get the the model that actually generated the data? Yes,
I was planning to run that too, but because it's taking longer, might not be able. But so now, as you saw earlier, let's say in this plot, that when you have the self-inhibitory modulation with the values that we're using, it's quite subtle in terms of the, let's say, neuronal activity effect. And so if I was to run this with the original value, which was 0.5, then that effect is not strong enough to justify this model, and the null model explains the data better. But now that I run it with 0.9, I think it should find the actual model as the best model for explaining the data.
Yeah. And what about the backward connection? Have you looked at modulating that? Is that distinguishable from, for instance, the forward connection?
Yeah, that's a good question. I haven't looked at that. I presume that this will depend on the magnitude of that modulation.
Yeah, so maybe that's something the attendees of the course could play around with, and see basically what kind of network configurations you can generate and which network configurations you can distinguish. In particular, from the null model, the benchmark model that we use in this case, that assumes no contextual modulation of the connectivity.
Yeah.
And of course, we haven't played around with SNR, but obviously in practice, what you would do, you would try to quantify an expected empirical SNR and use that in your simulations, and also kind of identify at which SNR level are the models no longer distinguishable from each other.
Yeah. All right. So the model fitting has finished, and so now we can rerun the model comparison for this new analysis. And as we can see now, the model 2 has the highest log model evidence, and that translates into very strong probability, or very high probability, for model 2 being the best model to explain the data, and that's the model with which we actually simulated this data. And as Andrea said, this is kind of something that you can play around with and find the limits where where things become no longer recoverable. But with that, I think that's probably all I had to share. Perhaps one thing I can mention, just as a clarification, here we set these, let's say, names for the regions, and we have a specific regions in mind, but really this whole exercise here is very general, and it would be exactly the same for thinking about any other 2 or more regions that we would simulate. And yeah, this example that we had in mind was just to make it, just give it some narrative and make it less abstract. But yeah, everything we've done can be applied for any kind of DCM analysis.
Yeah, and the anatomical prior knowledge has to do, of course, with the connectivity between the regions. But for certain regions, some of the parameters that govern the hemodynamic response might be slightly different. So, for instance, a lot of these parameters, their prior settings was based on studies using visual experiments and looking at visual or activations in the occipital cortices. But of course, if you're looking at modeling the effects of a changes in effective connectivity from a subcortical area like the striatum to a prefrontal area, you might have to consider differences in the timing of the hemodynamic response, particularly for sort of brainstem regions. There would be differences in the timing, and the way you could do that is through the estimation of these hemodynamic response model parameters such as kappa and gamma, but you might also have to specify a slightly different model that allows for that. First of all, does not put overly peaked prior distributions over those parameters, and allows these parameters to be estimated from the data. For example, when you're dealing with regions that have very different timings for the both signal. So I don't see any questions in the Ask the Question panel, but we can wait a couple of minutes if anything comes up. Otherwise, I think the plan is anyway to join GatherTown for a longer discussion with the whole group. Maybe I will just end with a note. We have this, there's this nice paper from Klaas and Stefan, which talks about 10 simple rules for a dynamic causal modeling analysis, which I recommend looking at and trying to figure out if DCM is the tool for you, if it's a tool to address your research questions. And if it is, I think it's important to consider the model or this technique into the design of your experiment. So to use these tools, such as what Povilas presented today, in order to simulate your hypotheses, essentially the cognitive network that you predict underlies your task, and to see basically what the limits of the model are before you actually collect the data. So I see there's a question from Reza. I wonder, would it be possible to assess the role of the nature of neurotransmission, whether it is receptor mediated or membrane diffusion in the modulation platform? So I don't know, Povilas, if you want to tackle that question.
Yeah, it's a tricky one. Not sure if I can answer it well.
Yeah, so I think this question could be addressed not with fMRI, but with EEG. And I think there is, and maybe not with convolution-based models, but rather conductance-based models. So there's a host of models called conductance-based models that have been used for EEG data or more invasive electrophysiological recordings. So I'm going to recommend you to look at the work from Rosalind Moran that has used these methods. Not fMRI, unless you perform an experimental manipulation where you alter neuromodulation, for example, with a pharmacological intervention. And even that is a bit problematic with fMRI because we know that it's going to change blood flow and, just due to the medication itself, not only because of the change in neural activity. So Erin actually posted a post-workshop survey here. Please fill it out. It would be great to get your feedback on the course, and just to have your feedback on the different sessions. That's really informative for us. And also Daniel posted a link to the paper. Yeah, this is one of the papers that I was referring to by Rosalind Moran that has actually looked at this more specifically using DCM. So with that, I think I'll invite you to join us at GatherTown. And again, want to thank Povilas and Peter for a great session. And thank you so much to everyone for your attention and for your questions. See you there.